In this section, we will be discussing about the replication and the cell mitosis. What is DNA replication? So, in DNA replication, there will be the duplication of DNA, which will last for 4 to 8 hours. And after this, there will be a phase of interphase that is for 1 to 2 hours. And after that, there is mitosis that lasts for 30 minutes. The DNA replication is semi conservative, which we will understand in a while. And both the strands will be replicated, not one of them like in transcription. So first of all, the DNA is winded, that is helical in structure. To unwind them, we need an enzyme, and the enzyme is called DNA helicase. Now, after this, we need to understand there is the direction of the DNA which it's running, that is 5 dash to 3 dash. On the right side, I have made 5 dash to 3 dash, and on the left side, that will be 3 dash to 5 dash. It will not be 5 dash together or 3 dash together. After the DNA helicase and unwind it, the let's say here is small part is still winded and here's some part is unwinded. So this part will be termed as the replication fork. Now I have made this portion over here the replication fork. After the formation of replication fork, like the RNA polymerase we had in the transcription, we will here have the DNA polymerase, which will add the new DNA strand in only 5 dash to 3 dash direction. So now, if the replication is starting from this side where I have marked the purple arrow, now we know that there is 5 dash to 3 dash only. Now it will create this, new strand of DNA will be created on the left side on the pink mark. So from here, new strand will be formed. This is 5 dash to 3 dash. This strand on the left side will be now called as the leading strand. And this strand on the other hand, that is the right hand, will be called as the lagging strand. In the lagging strand, this it will be not formed in the same pattern like it has formed in the left strand, but there will be small fragments. These small fragments will be like these. So these small fragments have been made. Now these have been made from this direction, 5 dash to the 3 direction, 5 dash to the 3 direction on the opposite end. So there are fragments of these. It has to be sticked together. To make them stick together, we have a glue inside them that is called the DNA ligase. Here, here will be the presence of DNA ligase, which will stick these fragments. These fragments are known as the Okazaki fragments. These fragments, Okazaki. Now, after some time, it has to stop. So, for stopping, it has exonucleases for the termination of this process, the replication of DNA. It will lead to termination. Now, also, one more fact that we know the DNA is made up of phosphate and the ribose, deoxyribose, sorry, and here is the gene present inside them. So even this these bonds can be broken. It can be broken by topoisomerase. This is the process of replication of DNA. And after that, I talked about the interphase. In interphase, there will be proof checking or the checking of the replication if there is mutations or not. That is, after the replication, it will lead to the transcription and translation, which leads to the formation of new protein. So if there is some abnormal protein presence, that will because of the incorrect gene replication, so that is the codons have not been correct, the anticodons are not correct, maybe in whatever reason. In this interphase, there will be proof checking of the replication phase. So now, after this, there will be mitosis. And before that, we should know there is chromosome replication as well. So what is chromosome? Chromosome is made up of DNA, and DNA was made up of gene. So chromosome is made up of DNA. It is surrounded on a protein. It is encircled on a protein called histone. So in the next slide, I have this. So these are the histone proteins. These are the proteins itself on which the DNA has been looped over itself. Now the many loops will be present, and this will be termed as the chromatin. and after chromatin, this is known as there will be supercoiling of the DNA, and supercoiling of the DNA will take place, and after that we will have a centromere in the center, which will lead to the formation of chromatid over here. It will lead to an X-shaped chromosome. And how many chromosomes are present inside a human body is 46 chromosomes. That is present in pair of 23. So in two pairs until 23 pairs. So these 23 pairs are present in such X format. That is, these are called the chromatid. The center is called as the centromere. The fourth one here is the long arm, and the third one here is the short arm. After this, we have the mitosis once again. So cell mitosis takes place for 30 minutes. This is where the cell replication happens. Now before every mitosis process, we should have a mitotic apparatus. To form the mitotic apparatus, we have a structure called centriole. Centriole looks like this, something which is cylindrical in shape. The size will be the length will be 4 micrometers and the diameter is 0.515 micrometers. These are paired. These are paired over here, as we can see. So here they will be joined at they are joined at the 90-degree formation. They are in the 90-degree formation. So at some point they will be joined. So these joint form at the point where they are joined is known as centrosome. Now in centriole, what really happens is due to centriole actually, they before the mitosis, the centrioles are moving apart. First, they are replicated. They are replicated during the interphase itself, or maybe just shortly around the replication phase of the DNA. And after that, they will separate from each other. Why they will separate? They will separate due to the polymerization of protein, microtubules, protein. Now here, in between the centrioles, there will be formation of spindle. This here will be termed as spindle, and on the other end of the centrioles, from the other side, they will form a circle spindles. These spindles will be termed as aster. Now they will lead to the formation of mitotic apparatus. Now after this, the cell mitosis takes place. That is of 5 phases: prophase, prometaphase, metaphase, anaphase, telophase. So first is the prophase. In prophase, there is condensation of chromosomes. Now the chromosomes have been replicated, the DNA have been replicated, everything has been replicated. So far they are still in the nucleus. From the nucleus, they are still being condensed in that X-shaped form. After that, there is also spindle formation going on from due to the centrioles. Then in the next phase, that is prometaphase, the asters which were on the sides of the centriole, they will attach to the chromatids. The X shape in the between, they had centromere, and on the long arms and the short arms were called the chromatids. So they are still bound at the prometaphase itself, and these asters which have entered into the nucleus, they will try to pull out. They will try to pull them, and centrioles itself will go to the cell poles, the upper pole and the lower pole. So here's one picture. This is the centriole, and here is the aster formation. And inside the nucleus, we have the nucleolus, that is lead to there is formation of the chromosome. In the B picture, the spindles have formed. The spindles have formed. Here is the aster. Then again, C is the prophase. So all A, B, C here pictures are the prophase. Now these have gone to the other poles of the cell. In a cell, it has gone to the upper pole or the sides of the pole at the very end. Now D is the prometaphase, and here in the prophase itself, there is condensation happening. So in the prometaphase, they have held together. They have held together at the centromere of the chromosome. Now these will be pulled at the center of the cell. That will be known as the metaphase. In the metaphase, they have been pulled towards the center. Now in the metaphase, they are being pulled because of molecular proteins. these are composed of actin or maybe myosin, which are also present in our muscles. So here in the metaphase, again, they are being pulled away. The chromosomes are being at the equicenter or exactly at the center, and centrioles have been pulled away to the one end of the cell, which can be due to the molecular motors. Now the next step is the anaphase. In the anaphase, they are ready to separate. Now there were 46 chromosomes in 23 pairs. Now, since they have replicated, so they were 46, after replication, there is 46 plus 46. So in the X format, if we see in the X format or the chromosome format, we will get 46 here and 46 here. So now, since they have started to separate, we can see that 46 chromosomes will go on one end and 46 daughter chromosomes will go on the one end. Here will be the telophase. After anaphase will be the telophase. In telophase, they have been separated to the centromere, the centrioles have pulled them to the one end of the cell. Now again in the telophase, they will be pinched. The cell will be pinched out and they will separate. The pinching is due to the microfilaments, which is composed of actin and myosin. So in metaphase, there was molecular motors, the lining of chromosome at the equatorial line. In the anaphase, the two chromatids are being pulled apart. So there is formation of 46 daughter chromatids. In the telophase, they have separated, everything has been separated, and the new cell formation has taken place. The new cell formation after the pinching, there is formation of the nuclear membrane and everything that has required or is required for the cell. For the control of the cell growth and cell reproduction, in the DNA itself, we have telomerase. These help while the cell is dividing, while there is mitosis going on, from the DNA to deteriorate. If these are not present, the DNA will be replicated as many times as the mitosis is going on, and this will lead to a cancer. And what in normal condition, when these are present, they can be deteriorated away. So this can be a sign of aging, the senescence. To get these back, we have an enzyme called telomerase. Here is telomere. Then for the cell size, it corresponds to the amount of function of DNA. To prevent mitosis, we have a chemical called colchicine. The cell size doesn't really matter how much it has to grow, it will grow. But the replication, the mitosis are really required. So that's all in this video.